How does X-ray diffraction work for DNA identification? Have you ever wondered how scientists can determine the structure of something as tiny as DNA? One of the key techniques they use is called X-ray diffraction. This method reveals the molecular structure of DNA by studying how X-rays scatter when they pass through DNA fibers. When a beam of X-rays strikes a well-ordered sample of DNA, the X-rays are diffracted in specific patterns. These patterns depend on how the atoms are arranged within the DNA molecule. By capturing these diffraction patterns on photographic film or detectors, scientists can infer the three-dimensional structure of DNA. The process starts with preparing DNA fibers in a controlled environment. Factors like water content are adjusted to stabilize different forms of DNA. When the X-rays hit these fibers, they scatter and create a pattern of spots or shapes. This pattern contains vital information about the distances between repeating units in the DNA and the overall shape of the molecule. One of the most famous examples of this technique is Rosalind Franklin's photo 51. This X-ray diffraction image of DNA's B form displayed a distinctive X-shaped pattern, indicating a helical structure. By analyzing this pattern, researchers could calculate important parameters like the radius of the helix, the distance between turns, and the angle of the helix. These measurements were essential in determining that DNA is a double helix made of two strands twisted around each other. Mathematically, the diffraction pattern can be understood through principles like Bragg's law. This law relates the angles at which X-rays are diffracted to the spacing between atomic planes in the crystal or fiber. The helical nature of DNA gives it a unique diffraction signature that is different from non-helical molecules. Scientists use these patterns to create accurate molecular models, confirming the arrangement of the sugar-phosphate backbone and the base pairs inside. Identifying DNA's structure through X-ray diffraction was a major milestone in science. It provided the physical basis for understanding how genetic information is stored and replicated. This method transformed biology by linking molecular structure to function. It also paved the way for advances in genetics, molecular biology, and medicine. In essence, X-ray diffraction works for DNA identification by directing X-rays at DNA fibers and recording the scattered rays as a diffraction pattern. Scientists interpret this pattern to reveal the spatial arrangement of atoms in DNA. They use mathematical models to translate the diffraction data into a three-dimensional structure, confirming the double helix form and the specific pairing of nucleotide bases. This technique was vital in the mid-20th century for uncovering the fundamental structure of life's genetic material. It marked a key innovation in the history of science and laid the groundwork for modern molecular biology.